All right, guys, so we are officially starting our very last unit, which is going to be Kingdom Animalia. We're going to focus on the different types of animals, just some commonalities among animals, and then we're also going to talk a little bit about some body systems and their functions, seeing as we are complex animals ourselves. So the first lesson is just going to kind of be about some just general characteristics of Kingdom Animalia. And so at this point, this is our last group. We've covered our prokaryotic groups, our bacteria and archaea. We then went on to cover Protista, which was kind of like the misfit kingdom of the eukaryotes. It's the eukaryotic organisms that don't fit either in fungi, plant, or animal. We then did Kingdom Fungi, and we last did Kingdom Plantae. And so we're going to finish off with Kingdom Animalia, which hopefully you do know that is the kingdom that we fall in. Now, what makes an animal an animal? Well, we have several specific characteristics that define animals. So they are multicellular, and multicellular animals. This is one of the things that sets us apart from like your protists that are animal-like, like an amoeba or a paramecium, and that animals must be multicellular. So that's why those unicellular organisms that may have animal-like qualities actually go in Kingdom Protista. Of course, we're in domain Eukarya. Now, most animals, the key is most, because what we're going to see all throughout the animal kingdom is simple to complex, simple to complex, simple to complex. We have very simplistic animals like a sponge, and if you looked at a sponge, you would never think that it was related to a human or an elephant or even a grasshopper, but you have very simplistic animals like sponges, but then you go all the way up to very complex animals like us and other mammals. And so for most, there's going to be a high degree of specialization. And we will be seeing this organization of life. We know that cells make up tissues, and most animals are going to have specialized tissues. There's a couple exceptions. And then those tissues will make up organs, which make up the organ systems. And so we're definitely going to see this organization in animals like we did in plants. Now, because of the multicellular, a lot of your animal cells have unique cell junctions. Like there are desmosomes and different types of junctions that hold our epithelial tissues together because our cells form sheets. And there's going to be junctions that you'll find in between muscle cells and things like that. Another thing that's kind of a defining characteristic of animals is that they are heterotrophs, but this time it's by ingestion. So remember, Kingdom Fungi were heterotrophic, but they absorbed. Animals are going to ingest in one way or another. Another defining characteristic is that when it comes to cell walls, they don't have them. Okay, animals' cells do not have cell walls at all. You know, we know that some of your prokaryotes had peptidoglycan, your fungi had chitin, your plants had cellulose. And so one of the things that sets animals apart is that they don't have one at all. So there is no cell wall, which means they are going to have some additional support in some way. We'll talk about that here in a little bit. And they are mostly sexual in their reproduction. There are some of your simpler animals that may be able to have asexual methods, and really even some of your complex may have ways to reproduce asexually, but we know that sexual is superior because of increased variation. And so for the most part, your animals are going to go through a life cycle where they produce haploid gametes. Those gametes are going to come together in fertilization, and the adult stage is going to be diploid. And so we're going to see this series of developmental stages in most of your animals. And so in terms of animal development, it starts with the zygote, which is the fertilized egg. So whenever the sperm fertilizes the egg, that forms a diploid zygote. This is a single cell. Every single one of you began as a zygote. And then what happens is very early on, that zygote, that one cell, is going to start to go through rapid cell division. But instead of dividing completely by cytokinesis, they actually kind of stick together. And so we call these cleavages. But the zygote's going to undergo rapid cell division. This is going to be mitotic division. So it's going to be making identical cells, identical cells, identical cells. So remember, every cell in your body has the same DNA. But what's going to happen over time is they will begin to specialize. So in general, though, you're just basically a blastula. You're a hollow ball of cells that are all the same. So imagine like a basketball, okay? That is what you looked like early on in development. You were just a hollow ball of cells. And then what happens is this hollow ball of cells begins to kind of pinch in on itself, as you see over here. So imagine kind of like a deflated basketball. That hollow ball begins to pinch in on itself in a process known as gastrulation. And this is what's essentially going to give rise to the beginnings of your digestive tract, okay? And so this opening here, where they begin to pinch in on itself, is known as the blastopore. And we're going to see here in several slides what's going to happen to that blastopore. Now, when it comes to classifying animals into groups, we have many different phyla under kingdom Animalia. And so how do we determine what group do they go in? Obviously, something like a jellyfish is going to be very different than something like a human or a fish or a reptile. Something like a grasshopper is significantly different from a worm. And so how do we put these organisms into different groups? Well, these are the characteristics that we're going to look at over the next several slides. So the first one we're going to look at is symmetry and body plan. Most animals are going to display some type of symmetry, but your simplest animals, your sponges, are what we call asymmetrical, which means they don't have a particular symmetrical body plan. There's no way you can kind of divide them in half to create equal sides. They just kind of have an irregular body plan. Some animals have what's called radial symmetry, and when you see the word radial, I want you to think of radius, and a radius always goes with a circle. So radial symmetry will be seen in animals that have like a circular body plan, like a sea anemone, like we see over here. The top of the jellyfish is kind of circular. Another thing that will fall under this radial symmetry are even things like sea stars, which I know they're not circular, but they have what's called a 5-point radial symmetry. And so there are multiple lines of symmetry that you could cut that starfish into. Now, most of your animals and your most advanced animals, for the most part, are going to be bilateral. And if we take a look at this word, bi means two, lateral means side. This word literally means two-sided symmetry. Now, for bilateral symmetry, there is only one line of symmetry. So imagine if you were going to cut yourself in two and have equal sides. The only way you could do it is to go straight down the middle of your forehead, through your nose, to the middle of your mouth, down through the center of your chest, abdomen, and all the way down, so that you have a left and a right side. So that's the key to bilateral, is that you divide it into two sides, left and right. Now, bilateral symmetry also gives rise to your other directional terms. And if you ever take anatomy in the future, you're going to get really comfortable with these. But you have things like ventral and dorsal, and then anterior and posterior. And so your bilaterally symmetrical animals are going to have sides to them. Something else you will see with bilateral symmetry is something known as cephalization. And if we break this word down as well, the word cephal, or this prefix cephal, refers to head. And so cephalization is when your sense organs get concentrated on one end of the body. Normally it's the front end. And so this is what's going to give rise to the head. If you think about something like a starfish, it doesn't have a head. Really, a jellyfish, it doesn't have a head, okay? So your earlier, simpler animals, they don't have a head of any type. And think about where the majority of your sense organs are. Your sight is in your head. Your smell is in your head. Your taste is in your head. Your hearing in your head. And so cephalization is just when you have these sensory organs that get concentrated on the front end of the body, and we will see heads or cephalization with bilateral symmetry only. Okay, if they have a radial type of symmetry or no symmetry, those animals would not have a head. And then the more complex you get, the more pronounced the cephalization. So here's an example over here. This is just your vertebrates, your fish, amphibians, your reptiles, birds, and mammals. But you can see how the development of that brain changes. And so like your mammals have greater cephalization than, say, a fish, okay? All right, something else that we could group animals by are their levels of organization. Now, most animals are going to be highly specialized, but not all of them follow that cell, tissue, organ. Okay, now we have some animals, the very simplistic ones, that they're pretty much only cellular. They don't really have organized tissues. They just have some specialized cells that help them accomplish some functions. Some animals, they have specialized tissues, but they don't have distinct organs. And we'll talk about this ectoderm and endoderm here in a moment. But your most complex animals are going to be highly specialized, and they're going to have very complex tissues and organ systems that allow them to accomplish the functions of life. Now, most of your animals are going to contain the four main tissue types, and we'll take a look at this again when we get to like body systems. But we have four main types of tissues. We have epithelial tissue, connective tissue, muscular tissue, and nervous tissue. And epithelial tissue is going to be like your coverings and your linings, like your skin. The outer layer of your skin is an example of an epithelial tissue. But all of your organs have epithelial tissue on the outside of them. And if it's a hollow organ, they're going to have epithelial tissue lining the inside of them as well. Connective tissues connect and support. There's a wide variety of connective tissues: bone, cartilage, fat. All of those would fit under connective tissue. Muscular tissue, of course, are going to be your muscles, and then nervous tissue is going to allow you to have a response type reaction. Now, I told you we would skip over that endoderm, ectoderm, and we cover that here in a minute. And so another way that we can classify animals is based on the number of germ layers. So this goes back to that animal development. So remember, you were a hollow ball of cells called a blastula, and then that hollow ball of cells begins to pinch in on itself in the process of gastrulation. You have that blastopore. And so what that is going to also give rise to are different layers of cells. So how is it that you go from this ball of cells with no specialization to a baby organism that has distinct structures and parts and functions? Well, a lot of it has to do with the positioning of the cells. Okay, so most animals are either going to be diploblastic, which means they have two of these three. It would be the ectoderm and the endoderm. Or a lot of your animals, your more complex animals, are going to be triploblastic, and these are the ones that have all three germ layers. Okay, so if you just kind of think about these prefixes here, we have ecto, endo, and meso. So ecto is going to be the outermost layer. This will become your skin as well as the nervous system. Endo means the inside or the inner layer. Now you're going to find the endoderm, it's going to form the inner linings of like your hollow organs, like your digestive tract, your respiratory tract. And then the mesoderm is going to be all the stuff in the middle. So meso is middle. So we're going to get all of our connective tissues like the bones, the cartilage, the blood, the fat. And then we're going to get all of our muscles from the mesoderm. So think about all the stuff in the middle. You have your inner digestive tube, you have the outer skin, and then the rest of it is mostly going to come from that mesoderm layer. And so here you can see where this hollow ball of cells kind of pinched in. And so this location and where these cells are located is going to help to tell them what they will be when they become specialized. Okay, it's very complex of how you go from this all the way up to a very complex organism with these specialized organs and systems. Another way that we can classify animals into groups is based on their body cavity. Now, what a body cavity is it's usually a space between the digestive tract and the outside of the body. We call that a coelom. That's another name for it. Normally, it's filled with fluid, and we've got cavities inside of us, and they cushion our organs. They kind of help allow them to kind of move around a little bit on their own and adjust. And so we can put animals in one of three groups. We have acoelomates, which anytime you put an A or an an in front of something, it means not or no. So acoelomates don't have a body cavity at all. And we'll see these as your flatworms. You can see it's tissue on tissue on tissue. There's no white space here. It's just your digestive tract. You have the middle layer, the mesoderm, and then you have the outer layer from the ectoderm. So there is no cavity or space in between the tissues. Then we also have pseudocoelomates, which pseudo means false. So this is a fake cavity. So you will find this in your roundworms. And so here we have a cavity, but you see how the red, the mesoderm tissue is only on the outside. So it's not fully lined with that mesoderm, and so we call it a fake cavity. And then a lot of your animals are going to be coelomates, which this is where they have a fully lined body cavity. The mesoderm is completely surrounding it inside and out. And we see this as early as our segmented worms, all the way up through, up into us, a lot of your invertebrates as well as your vertebrates. Another way that we can classify animals is based on segmentation, and this is where you have repetition of body parts. We start to see this with the earthworms. In fact, they're called segmented worms. You see it in the arthropods, which are like your insects and your crustaceans. They have segmented bodies. And then we also see it in your vertebrates or your chordates. So think about your vertebra. It's a repeating series of bones, and we can group those into sections. And so this segmentation allows for improved movement, better able to regenerate for those animals that can regenerate. And then we also have these specialized body regions. And then one of the last things we can look at in terms of that list, that original list of 7, is the animal a protostome or a deuterostome? And this is going to go back to that blastopore. So in triploblastic animals, which means they have all 3 germ layers, they will either be classified as a protostome or a deuterostome. Now, the blastopore is that initial opening, okay? So whenever we have that gastrulation happening, we're forming those different germ layers. That opening, when we had that pinching in, is called the blastopore. Now, if it's a protostome, that blastopore is going to become the mouth. That's going to be the mouth end of the digestive tract. And it also has some special type of cleavage called spiral indeterminate. What that means is that very early on, these cells know what they're going to be, okay? Now, deuterostomes, which includes us and echinoderms, the deuterostomes, that blastopore develops into the anus. And so you kind of think about the word deutero, what that may sound like, and then you can connect that to anus, and that will help you remember it. But they have what's called a radial indeterminate cleavage. So early on, these cells don't know what they're going to become just yet. That'll be determined later on in development. There's also some differences in how their coelom forms based on like the mesoderm and things like that. All right, a couple other things about animals. Most animals are motile. Now, this is a little bit different than the word mobile. The key to motile is they're able to move independently on their own using energy from metabolism. And so for the most part, most animals can move around on their own. There are some that are sessile, like the sea anemones that you see up here. Coral would be sessile. A lot of your polyp stages are what we call sessile. But for the most part, most animals are able to move around with some type of muscle tissue. Now, usually, though, if an animal is able to move, they're going to need some type of support. Like even us, our muscles would be useless without our bones. And so there needs to be some type of support system. So animals could have hydrostatic skeletons, which is kind of like a water skeleton. It's a fluid-filled cavity. They could have exoskeletons like your insects, or a crab, where they have that hard outer shell or outer body covering. That's actually made of chitin, the same stuff that you'll find in fungal cell walls. Or they could have endoskeletons like what we have, where you have the bones located on the inside, or some kind of hard structures on the inside. We could also look at animals in terms of thermoregulation. For the most part, all animals have to maintain a somewhat tolerable range of temperature, but we can group them as either endotherms or ectotherms. Now, these are the more appropriate terms for what you probably have heard in terms of warm-blooded or cold-blooded. Now, cold-blooded really isn't a great description because it kind of makes you assume that their blood is cold. But the cold-blooded is the word that goes with your ectotherms. And what it is, it's not necessarily that their blood is cold, but their body temperature changes by the environment. So if it is colder outside, their body temperature is lower. If it's warmer outside, their body temperature is higher. And what you'll see in ectotherms, which is really quite a lot of animals, most of your animals are really probably ectothermic, but what you'll do is you'll see them kind of adjusting to the external temperature. So if it's not super, super hot outside, you'll often find them like basking in the sun. If it gets too hot, though, they'll usually burrow down into the shade, or they'll get in areas where it's not as hot. They may change color to absorb, less light. They may change location of where they are in the water to get closer to warm or more to cold, depending on the situation. And so a lot of times they have these behaviors that allow them to kind of either be cooler or warmer, because they can't get too hot or too cold or they would die just like any other organism. Now, endotherms, though, have a constant temperature. We are endotherms, and so are birds, mammals and birds. And so if it's cold outside, that doesn't mean that our bodies are cold. We have mechanisms that help us maintain a relatively constant body temperature. For us, it's around 98.6 degrees Fahrenheit or 37 degrees Celsius. And if we go too high above or below, we could be in some serious trouble. So we have very high metabolism to help us generate heat in our bodies. We have insulation. You could have hair or fat. We've got negative feedback mechanisms, like when we get hot, we sweat, or when we get cold, we shiver. All of those are helping us maintain that fairly constant temperature. So it's really not necessarily a cold-blooded versus a warm-blooded. It's more of a constant temperature versus a variable temperature. And the very last thing we're going to talk about here are our different animal groups. And then as we continue on as we move forward, we're going to go into more details on these animal groups. So the majority of animals, over 95%, I saw where a lot of times it's more around 97%, but we're just going to say over 95% are what we call invertebrates. And invertebrates are animals that do not have a backbone. They're very diverse. They range from sponges to worms to crabs to insects to shells, things like mollusks, clams. And so you have a very wide variety here. There are some more phyla that I don't have listed here, but these are your most common invertebrate phyla. We'll take a look at those moving forward. Here's kind of a phylogenetic tree of all the different ones. So you can see some of these traits that we mentioned, like bilateral symmetry, radial symmetry, protostomes versus deuterostomes. And then the other animals are what we call chordates. And so in order to be considered a chordate, at some point in their life, these four characteristics had to be present. A lot of times these are present in the embryo stage. But you have a dorsal hollow nerve cord. This is what will essentially become the spinal cord, that major nerve line. The notochord is like a rod, like a protective rod that kind of protects that dorsal hollow nerve cord. And this is what will become the kind of the vertebra that protect around it. You have the pharyngeal or pharyngeal clefts or slits. In some organisms, these will become gills. They actually become part of our inner ear. And then there's what's called a tail or a post-anal tail. Now, obviously, we are chordates, but we don't have tails currently. So they just had to have these at some point in their life. Now, within the chordates, there are two groups. There's the non-vertebrate chordates, which means they don't have true vertebra, but they do have these four characteristics here. And there's only a couple examples. It's the tunicates and the lancelets. They really look more like an invertebrate than they do a vertebrate, or the chordates. And then the majority of your chordates are going to be vertebrates. And these are your animals that have backbones, your fish, amphibians, reptiles, birds, and mammals. And so there you can see the different groups that are present within your chordates. And that is going to wrap up our first unit on, or our first lesson on Kingdom Animalia. As we move forward, we're going to be taking a look at these groups of animals more closely, doing some lab activities and a couple dissections, and hopefully you guys will just finish out strong. We are there. We are almost there, y'all. Just keep working. Hope to see you soon, and of course, if you have any questions, just let me know.